Lesson 17: Ingestion of resistant bacteria producing human disease. Aquaculture, the practice of cultivating aquatic organisms for food, has seen tremendous growth due to increasing global demands for fish and seafood. However, this growth is accompanied by significant public health challenges, particularly concerning the ingestion of antibiotic-resistant bacteria from aquaculture environments, which can lead to human diseases. 17.1 The issue of antibiotic resistance in aquaculture. Antibiotics are extensively used in aquaculture to prevent and treat bacterial infections in fish. This widespread use, often in subtherapeutic doses, creates a conducive environment for the emergence and proliferation of antibiotic-resistant bacteria. These bacteria can be pathogenic to humans, and their presence in the food chain represents a critical public health concern. 17.2 Mechanisms of resistance development. The development of antibiotic resistance in aquaculture environments occurs through several mechanisms. Selective pressure. Continuous exposure to antibiotics exerts selective pressure on bacterial populations, favoring the survival of resistant strains. Horizontal gene transfer. Resistant bacteria can transfer resistance genes to other bacteria through mechanisms such as conjugation, transformation, and transduction. Mutation. Spontaneous genetic mutations can confer resistance to antibiotics, which is then propagated within the bacterial population. 17.3 Human health implications. The ingestion of resistant bacteria from aquaculture products can lead to various human diseases. Pathogens such as Escherichia coli, Salmonella spp., and Vibrio spp. Are known to cause severe gastrointestinal infections. These infections become particularly problematic when the bacteria are resistant to multiple antibiotics, complicating treatment and leading to higher morbidity and mortality rates. 17.4 Transmission pathways. Humans can ingest antibiotic-resistant bacteria through several pathways. Consumption of contaminated fish. Eating undercooked or raw fish that harbors resistant bacteria can directly introduce these pathogens into the human body. Cross-contamination. During food preparation, bacteria can transfer from contaminated fish to other foods or surfaces, leading to indirect ingestion. Water contamination. Aquaculture environments can contaminate water sources with resistant bacteria, which may then be ingested through drinking water or recreational activities. 17.5 Mitigation strategies. To reduce the risk of human disease from the ingestion of resistant bacteria, several strategies can be implemented. Prudent use of antibiotics. Limiting antibiotic use to necessary cases and avoiding subtherapeutic doses can reduce selective pressure on bacterial populations. Alternative treatments. Utilizing probiotics, and vaccines can help manage bacterial infections without promoting resistance. Enhanced monitoring. Regular surveillance of antibiotic resistance patterns in aquaculture environments can facilitate early detection and intervention. Regulatory measures. Implementing stringent regulations on antibiotic use in aquaculture can help control the spread of resistant bacteria. Public awareness. Educating consumers about the risks associated with consuming raw or undercooked seafood and proper food handling practices can reduce the incidence of bacterial infections. *Glossary 1. Antibiotic resistance. The ability of bacteria to survive and multiply in the presence of antibiotics that would normally kill them or inhibit their growth. 2. Selective pressure. The environmental factors that favor the survival and reproduction of resistant bacteria over susceptible ones. 3. Horizontal gene transfer. The transfer of genetic material between bacteria through mechanisms such as conjugation, transformation, or transduction. 4. Mutation, a change in the DNA sequence of an organism that can lead to antibiotic resistance. 5. Pathogen, a microorganism that can cause disease. 6. Gastrointestinal infections, infections that affect the stomach and intestines, often causing symptoms like diarrhea, vomiting, and abdominal pain. 7. Probiotics, live microorganisms that provide health benefits when consumed, often by improving or restoring gut flora. 8. Probiotics, substances that induce the growth or activity of beneficial microorganisms such as bacteria and fungi. 9. Surveillance, the systematic collection, analysis, and interpretation of health-related data to plan, implement, and evaluate public health practices. 10. Cross-contamination, the transfer of harmful bacteria or substances from one food product to another, often through direct contact or via utensils, surfaces, or hands.